Hey everyone, this is Vaibhav Chandelier from ESIC Medical College, Hyderabad, and today we'll be talking about another bloodstream infection topic, which is brucellosis, caused by the organism Brucella. So let's start with the introduction. So this Brucella, it is a gram-negative Coccobacilli, okay, and it is an obligate aerobe, gram-negative Coccobacilli, obligate aerobe, and it is of multiple types based on the primary organism it infects before the man. Okay, so if it infects the sheep and goat first, that is Brucella melitensis, dog Brucella canis, cattle Brucella abortus, and pigs Brucella sui. Out of this, the most pathogenic, which most commonly infects man, is Brucella melitensis, which is primarily what we will focus on. But the other forms may also infect man. Coming to the pathogenesis, what is the source of infection? How do we get this infection? Basically, a contaminated animal. The organism will go from that animal to the human via abraded skin or mucosa. This is the most common route, abraded skin or mucosa. Otherwise, there can be inhalation of aerosols in infected animal shelters, any cow shelters, or wherever you keep these infected animals, there can be inhalation of aerosols. Unpasteurized milk or undercooked meat of these said infected animals can cause the infection, and very rarely person to person contact via breast milk or blood transfusions can cause the infection, but again that is very rare because you do a lot of tests before doing that. So after all these things, there is either inoculation, ingestion or inhalation of the infected organism, the brucella, into the body of the human, of the man, either by inoculation, ingestion or inhalation, where the organism then enters the blood via hematogenous spread. Okay, so there is hematogenous spread. Where do they spread? The reticuloendothelial system, which is the lymph nodes, bone marrow, spleen or liver, any of these places. And since it is infecting the body in all these places, there will be a neutrophilic infiltration to fight off the infection. This neutrophilic infiltration will lead to a chronic inflammation, and a chronic inflammation usually ends in granuloma formation. And wherever granuloma formation occurs, that is a very important point which you should remember. So granuloma formation occurs here, right? Coming next, this organism is a facultative intracellular organism whose main virulence factor is its LPS antigen, the lipopolysaccharide antigen in its cell wall, which helps it survive. This LPS antigen, if you want to remember, has two types, M antigen and A antigen, although this is not very important, but this is the main virulence factor. Coming to how the man will respond. The host response is triggering of the cell-mediated immunity, which leads to Th1 response, leading to IFN gamma release, and which finally kills the bacteria. Okay, so this is the overall pathogenesis. Coming to the clinical manifestations, here we see a classical triad of fever with night sweats, hepatosplenomegaly, and arthralgia or arthritis. This is a triad you should remember. Fever with night sweats, hepatosplenomegaly, arthralgia or arthritis. The triad of brucellosis. Next, there is a less severe typhoid-like illness which is possible. Typhoid-like illness, because we are ingesting undercooked meat and water and all that. It can occur by that source also. So less severe typhoid-like illness. There is a characteristic, the most important factor in this whole mind map, probably undulating fever, which means it is a remittent course of fever, remittent course, which means there will be afebrile periods between weeks of febrile period. Okay, you'll have maybe 1 or 1.5 weeks of fever, followed by few days of afebrile period, then again fever. That is what undulating fever is. If you want to remember an extra point, this is also known as Malta fever. Okay, but mainly all you have to remember is the word undulating fever for brucellosis. You also get other conditions like osteomyelitis, severe arthritis, weakness, weight loss, anorexia, or CNS symptoms like depression and lethargy. Right? That was the clinical manifestations. Remember the triad: fever, night sweats, hepatosplenomegaly, arthralgia, arthritis, or undulating fever. This is the two most important facts in clinical manifestations. Finally, coming to the lab diagnosis, the specimen can be taken from blood, bone marrow, or CSF, right? And you can culture the organism where Castaneda biphasic BHI broth or agar. Castaneda's biphasic broth or agar, BHI broth or agar. Then you can also do a automated backtech or backt alert, all these things, just the usual stuff. Culture smear and motility testing will show, as we know, gram-negative Coccobacilli as we discussed, and this organism is non-motile. How do we know that? We do a hanging drop test, which shows non-motile organism. Coming to something called the milk ring test, I don't think it's done anymore, but still important to know the milk ring test. You take a drop of colored Brucella antigen. Okay, you take a colored Brucella antigen drop. It is added to the milk in a test tube. So you add this drop to milk in a test tube, incubated at 70 degrees Celsius for 40 to 50 minutes. And what you see, if positive, that is, if Brucella antibody is present in the milk, you see a blue ring at the top of the test tube and the milk is unstained. While if it is a negative test, that is means no Brucella, there will be no ring and the milk becomes uniformly blue. Correct? Now coming to how will you identify? Identification will be done via Maldita or Vitek, and otherwise if you don't want to do this, biochemical means are catalase positive and oxidase positive organism. Okay, easy. Antibody detection, you can always use ELISA, but here we use a characteristic standard agglutination test, SAT. Basically, we're trying to detect antibodies against LPS antigens of Brucella. Remember, we talked about the virulence factor LPS antigen. We're trying to detect antibodies against that. So this standard agglutination test is basically a tube agglutination test, which if positive, how do we know this test will be positive? There will be high titers of these antibodies, said antibodies we discussed. There will be high titers. You don't have to remember the values, but if this is positive, this either means we either have an acute infection or chronic infection of Brucella. Acute how? We know that IgM antibodies whenever detected means acute, and IgG indicates chronic, right? So it can tell us that there is an acute or chronic infection, but standard agglutination test does not differentiate whether, it cannot tell us if it is acute or chronic. It can just tell it is either of these. To differentiate between the two, we club this standard agglutination test with 2-mercaptoethanol testing. Now, if we add 2-mercaptoethanol, this will destroy the IgM antigen. And if it destroys the IgM antigen, what we're left with is IgG antigen and IgG antibody, sorry. And a positive test means chronic brucellosis because now IgM is destroyed, so IgG is left. And a negative test means IgM positivity means it's acute brucellosis. Okay, that's all. The 2-mercaptoethanol will destroy the IgM, leaving behind the IgG, indicating a chronic brucellosis. That's it. Molecular methods are PCR and biofire film array. That's it. So this is the lab diagnosis. And the treatment will be rifampicin and doxycycline, 6 weeks treatment. But the names are enough to remember, rifampicin and doxycycline. Quick recap, this is a gram-negative Coccobacilli, and the type most commonly infecting humans is Brucella melitensis, and the source of infection most commonly is contaminated infection of the animal into abraded skin or mucosa, otherwise through unpasteurized milk or undercooked meat, and then it goes into the reticuloendothelial system where it will trigger neutrophilic infiltration and finally lead to granuloma formation. LPS antigen is the virulence factor, and there is triggering of our cell-mediated immunity leading to IFN gamma and Th1 response which kills the bacteria. Clinical features very much important: classical triad, fever, night sweats, hepatosplenomegaly, arthritis, arthralgia, and undulating fever, very important. Afebrile periods with weeks of in between weeks of febrile period. Lab diagnosis, what was important? Castaneda biphasic BHI broth or agar is used for culture. Apart from that, non-motile hanging drop test, and you can do a classical, you have to remember what standard agglutination test is, and this will not differentiate if it's acute or chronic. For that, you have to add 2-mercaptoethanol to differentiate if it is acute infection or chronic. And yeah, that is it for the lab diagnosis, and that is it for brucellosis. And yeah, see you guys in the next video. Thank you for watching.